Dr. Jennifer Malcolm completed her degree in microbiology at Glasgow Caledonian University, where she first sparked an interest in host pathogen interactions in health and disease. She pursued this interest during her PhD at the University of Glasgow School of Medicine, Dentistry, and Nursing, where she identified how oral biofilm development in young children is accompanied by salivary immune responses. Following her PhD, she worked as a postdoctoral research associate at the University of Glasgow School of Infection and Immunity. And here she identified novel therapeutic targets for the treatment of periodontitis and began her work investigating mechanisms driving autoimmunity in rheumatoid arthritis. And in 2018, she took up a teaching position at the University of Glasgow Dental School, where she's exploring how oral bacteria exploit innate immunity to subvert adaptive immune responses and how this impacts the maintenance of immune tolerance in the oral mucosa. And her presentation is titled C-terminal citrullinated peptide alters APC-CD4 T cell interactions leading to breach of immune tolerance. Jennifer?
Great, thanks so much for the introduction. So yeah, today I would like to share some data with you showing how C-terminal citrulination can breach immune tolerance as a potential mechanism for triggering autoimmunity in people with rheumatoid arthritis. And I have to apologise because the window cleaners have just arrived at my house, so hopefully they won't disrupt me too much and you'll be able to hear what I'm saying. So I just would like to begin by outlining that citrulination is a post-translational modification that is catalysed by enzymes known as peptidoarginine deaminases. And in humans, we express 5 different isoforms of these enzymes, and each have different cell and tissue-specific expression, but they each essentially catalyse the same reaction, and that is that they deaminate positively charged arginine to neutrally charged citrulline. And this occurs in the concentration, a high concentration of calcium. And so this change in the charge of the proteins then can modify the protein structure, function, and potentially their immunogenicity. But it is important to note that in health, citrullination is not overtly immunogenic, and indeed it has important cell and tissue-specific physiological functions. But in RA, autoimmunity to citrullinated proteins, so it's highly specific for the development of rheumatoid arthritis. And so in RA then, preceding the onset of the disease is an immunological triggering event that causes our adaptive immune cells to respond to citrullinated self-antigens. And this breach of immune tolerance is best evidenced by the emergence of autoantibodies that are specific for citrullinated proteins in patient serum. And we refer to these autoantibodies as ACPAs, and indeed seropositivity for ACPAs is diagnostic for RA. I would also like to point out that autoreactive CD4 T cells specific for citrullinated epitopes can also be found in this very early stage of the disease, and we think they're really important for driving the autoimmune response, but we're not exactly sure of their antigen specificity, and so these cells are much harder to study at these very early stages. But we do know that over time, the autoreactive T and B cell response and the autoantibody response matures, and then at some point, for reasons that we don't really fully understand, is it starts to infiltrate and target our synovial joints. And it is at this stage that joint inflammation becomes clinically apparent. Now, it is possible to halt the progression to chronic RA if early and aggressive immunosuppressive therapies are given at this early RA stage. However, the majority of patients do have ongoing autoimmunity. Relapses and flares are common, and they often require to take immunosuppressive therapies, possibly for lifelong. So ultimately, our goal is to restore immune tolerance and to cure the disease. And currently that remains very much the exception rather than the rule. And I think if we want to make that the rule for all patients, then one of the things that we really need to better understand are these triggering events and what causes this breach of immune tolerance in the first place. So what do we know about this triggering event? Well, like many autoimmune diseases, we know that RA develops in a background that is dominated by particular HLA-alleles, and in particular these HLA-DR alleles, which encode an amino acid motif known as the shared epitope. And this shared epitope has been shown in some cases to present citrullinated epitopes more efficiently than the native arginine-containing epitopes. And we know that risk of RA is significantly elevated where there is an interaction between these HLA-DR alleles with certain environmental factors such as smoking or microbial dysbiosis, which drives chronic inflammation at mucosal surfaces. So this strong environmental component, and together with this observation that the autoimmune response occurs or is detectable before the onset of joint inflammation, strongly suggests that the triggering event for rheumatoid arthritis occurs outside of the joint. And epidemiological studies and increasingly experimental data indicate that these sites of disease triggering are likely to be the lungs of smokers or the gums or guts of patients with microbial-driven inflammation. So what we think is happening is that at these surfaces, you're getting post-translational modification of antigen and generation of citrullinated neoepitopes, which are presented by these HLA-DR molecules to autoreactive CD4 T cells and B cells, which drive the emergence of APC in serum, which later targets the joints. And so at this point, I just want to highlight that what I'm specifically interested in is understanding how bacterial-driven inflammation in our gums might drive the activation of autoreactive CD4 T cells. And so in line with that, I'm particularly interested in the bacterial pathogen Porphyromonas gingivalis, which is perhaps best known for its association with periodontitis, which is a chronic inflammatory condition whereby the tooth-associated biofilm drives chronic inflammation, which destroys the connective tissues that surround and support our teeth. But with particular relevance to RA are two of P. gingivalis' virulence factors. So one of these are its arginine gingipanes, and the other is its own peptidoarginine deaminase. So P. gingivalis is the only known microbial species that expresses its own PAD enzyme, and we refer to this as PPAD. So these arginine gingipanes, these are cysteine proteases, which are capable of cleaving host and microbial-derived proteins at arginine peptide bonds. And so they give rise to peptides with C-terminal arginine residues. PPAD then preferentially citrullinates those C-terminal arginines, giving rise to C-terminal citrullinated epitopes. And so I do want to point out as well that host pads are implicated in RA pathogenesis, but the way in which they citrulinate proteins is quite distinct. So host pads will preferentially citrulinate internal arginine residues in a process which I've already alluded to is highly calcium dependent. So our working hypothesis then is that infection with Porphyromonas gingivalis generates bacterial and host-derived C-terminally citrullinated neoepitopes, which are capable of triggering autoreactive CD4 T cells. So the challenge for us then is to find these autoreactive T cells that recognise these C-terminally citrullinated host-derived peptides. And of course, looking for antigen-specific T cells can be a little bit like looking for a needle in a haystack. But of course, with the right tools, it is possible to find the needles, things like MHC tetramers and perhaps even anchorons. And, you know, I've alluded to the fact that we don't really know what are the inciting antigens for the autoimmune response in RA, and we don't really know either what, in vivo at least, how P. gingivalis, what antigens it C-terminally citrullinates. And perhaps we should have tried the pro-immune antigen presentation assays, but we didn't do that. We took a different approach, and we instead asked, does an antigen-specific T cell care if its cognate peptide is C-terminally citrullinated? And for this, we used the OTII T cell, which expresses a T cell receptor, which specifically recognises peptide over in complex with class 2. And you'll notice here that the peptide over sequence, it has a C-terminal arginine residue. And rather than asking P. gingivalis to do it for us, we instead used has proimmune to synthesize us the citrullinated version of the peptide. So using this system then, we first wanted to ask, can our OTII T cells recognize and respond to the citrullinated peptide? And so using an in vitro assay, we were able to show that at early time points, OTII T cells proliferate equivalently to both peptides. But when we looked at later time points, we consistently observed this small but statistically significant increase in proliferative capacity in response to our citrullinated version of the peptide. And when we looked at this in a little bit more detail, we observed that this enhanced proliferation was preceded by higher levels of IL-2 in cell culture supernatants, and it coincided with sustained expression of the transcription factor c-Myc. And c-Myc has been shown to correlate TCR signaling strength with the size of the proliferative response. We also observed that our OT2s responding to the citrullinated peptide expressed higher levels of activation markers, CD25, and higher levels of CD71, and that they released significantly more interferon gamma into our cell culture supernatants. So we then wondered whether there might be a difference in the strength of physical interactions between our antigen-presenting cells and the OTII T cells in the presence of either peptide, because we know that TCR affinity for peptide MHC complexes often translates into differences in the physical interaction between the cells. And so to measure this, we used an optical trapping system, which uses lasers to measure the force required to pull apart interacting cell pairs. So I just play this video for you. So this is our OTII T cell here, and it's interacting with a dendritic cell here. And after a period of time, we apply the laser just off to the side and gradually increase the strength until it's sufficient to pull those cells apart. And so using this technique, we can measure changes in the force of interaction required to pull apart APCs and T cells in the presence or absence of peptides. And so we applied this system to ask whether we could measure a difference in the strength of interaction in the presence of P over or P over CIT. And you can see here, like in the previous data, when in the presence of the peptide, significantly greater force is required to pull apart the interacting cell pairs compared to the absence of peptide. And when we look at with our citrullinated version, even more force was required to pull apart those interacting cell pairs. So this suggests that with the citrullinated peptide, the interaction between antigen-presenting cells and OTII T cells is physically stronger. So we next wanted to look in vivo to see if we could recapitulate some of our in vitro data. So here we transferred CFSE labeled OTII T cells into recipient mice, and then we IV immunized them with either peptide in LPS or PBS control. And when we looked in the draining lymph nodes of the mice, we could see that there were significantly more OTII T cells in the mice immunized with the citrullinated version of the peptide. And then when we looked at the CFSE divisions on those cells, we could see that more OTII cells had undergone more rounds of cell division in response to the CIT peptides compared with the native peptide. So having established that in the context of immune priming, our OTII T cells responded to P over CIT with enhanced proliferation, we next wanted to know what would happen in the context of immune tolerance. So can C-terminal citrullination override immune tolerance? And so here we in vitro generated some tolerogenic or LPS matured bone marrow DCs, and you can see that they expressed equivalent levels of CD11C, but our tolerogenic bone marrow DCs express lower levels of class 2, CD86, CD80, CD40, and they released lower amounts of IL-12 and secreted more IL-10. And so using these cells, we peptide pulsed them with either peptide and then co-cultured them with CFSE labeled OTII T cells. And you can see here that in response to co-culture with mature bone marrow-derived dendritic cells, this OTII T cells underwent several rounds of cell division to either peptide, whereas with our tolerogenic bone marrow DCs, they did not induce substantial cell division to native peptide, but they did with the citrullinated version of the peptide. And actually, this was accompanied by quite high, robust expression of activation markers CD71 or CD25, which was not evident with the tolerogenic bone marrow DCs presenting the native peptide. And so together, these data really suggest that OTII T cells responding to the citrullinated version of our peptide were less dependent on co-stimulation to drive proliferation and activation. And so we next wanted to know whether C-terminal citrullination was sufficient to breach immune tolerance to native antigen in vivo. And in order to establish a sort of functional output of T cell tolerance, we combined an oral tolerance model to ovalbumin, combined that with a delayed type hypersensitivity model. So following transfer of OTII T cells into a recipient mice, we induced oral tolerance by feeding the mice ovalbumin in their drinking water for a period of 10 days. This was followed by systemic immunization with either peptide and CFA. And then we measured the delayed type hypersensitivity response by measuring foot pad swelling following challenge with heat aggravated over in the foot pad. So where our mice received ovalbumin in their drinking water, we refer to these as our tolerized mice. And where they did not receive ovalbumin in their drinking water, these are our primed mice. So we can see here that in mice that did not consume ovalbumin in their drinking water, systemic immunization with peptide ova, followed by HAO on the foot pad, induced substantial paw swelling relative to their contralateral control paws. And in comparison, the mice who did receive ovalbumin, so our tolerized mice, they had significantly less paw swelling. So these data suggest that, you know, we did have a functional model of tolerance, and feeding ovalbumin induced that tolerance. So, but when we looked at systemic immunization with our citrullinated peptide, we can see that regardless of whether they had ovalbumin in their drinking water, they developed significant paw swelling. And in our tolerized P/CIT mice shown here in purple, the paw swelling was significantly greater compared with our tolerized mice with the native P/OVA. So these data demonstrate the capacity of C-terminal citrullination to breach immune tolerance in vivo. And so then I'd just like to summarize then. So I've shown you today how a T cell receptor can have cross-reactivity between both its cognate and C-terminal citrullinated epitope. I've shown how C-terminal citrullination was able to alter the intensity of antigen-presenting cell CD4 T cell interactions, and how this altered antigen-specific T cell recognition and their functional responses in the context of both immunity and immune tolerance. And so we think this shows the capacity for C-terminal citrullinated epitopes to breach immune tolerance as a potential mechanism to trigger autoimmunity in rheumatoid arthritis. So I would just like to finish then by acknowledging everyone who was involved in this study. And thanks for listening.
Great. Thank you, Jennifer. Looks like Jeremy has a question. Please go ahead.
Yeah, thanks very much, Jennifer. That was really great. And a lot of work there over a lot of years. I appreciate that it's a story that kind of develops over time. And you also mentioned about you mentioned about the sort of the ability to track these antigen-specific T cells using tetramers. Have you also done that as well as using the peptide? Have you looked at the citrullinated modified peptide in the context of IAB tetramer?
No, so we haven't done those experiments. I mean, we decided to use OVA as a model antigen system. Ideally, if we knew what the antigens we wanted to look for, which citrullinated antigens are driving this autoimmune reaction, I mean, that would be ideal. Actually, we are doing some experiments just now just to work out how P. gingivalis and how P pads might be operating and to making these cells. And actually, perhaps something like your ProPresent assay could be really useful in that regard.
Yeah, it's an interesting one because we've certainly applied sort of bacterial lysates to the context of ProPresent. So with that bacteria, we can actually load the dendritic cells, and obviously you can have a whole range of different HLA types, and we can select DR4, I guess, which is obviously associated with RA and other alleles as well. So we can select donors with that type, and then identify. You're not necessarily interested in the specific peptides, but you are of course here, but it will also identify specific antigens that might be more
Yes.
prone to citrullination than others. And because in ProPresent, obviously the mass is a little bit different with the citrullination, we can specifically identify citrullinated modified peptides. As long as we know exactly which part of that protein we're looking for, we can actually search for those individual modified arginines. So yeah, it can definitely be done in that
Yeah,
context.
that sounds really interesting. I think it's worth exploring. Yeah, thanks.
No, thank you too. It's great.
All right, are there any other questions? Okay. Great, thank you, Jennifer.